Okay, folks, it's been a long and tedious morning, but I think I've got some good results and I'm excited to share them with you. Let me tell you what I did. I did a titration very similar to the one we did in our laboratory, but this one was more controlled. And let me tell you what I did. I first of all took a hydrochloric acid solution. This was a 1 molar hydrochloric acid solution. I started with 1 liter of that 1 molar hydrochloric acid solution. And I titrated it with a base from the burette. So the base was a 1 molar sodium hydroxide solution, and I carefully determined the pH using a pH sensor connected to a LabQuest. So this is the pH sensor that I used, and what I did was, first of all, determine the original pH of the hydrochloric acid, and I'll give you that data in just a minute. But I took the original data, and then what I did was slowly I opened up the stopcock and I released 1 milliliter of sodium hydroxide into the flask, Erlenmeyer flask, and then closed it back up again, determined the pH, recorded that value, and then did that again many times until I created a titration curve. And I want to discuss that titration curve with you. That's really the basis of this lesson. So let's talk about a little bit about what's happening in this titration. So remember again that what I did was I titrated a strong acid with a strong base. Now, at the very beginning, I took the pH, as I've told you, I took the pH and I determined that the pH was 0. Now that makes sense. That makes sense because of the fact that, remember, that I started out with a 1 molar solution, and we now know that pH is equal to minus the logarithm to the base 10 of the hydrogen ion concentration, So, if that's true, and I know the hydrogen ion concentration for HCl is 1 molar, then I also know that the hydrogen ion concentration is 1 molar as well. So if I say that's equal to minus the log of 1, well, what's the log of 1? It's 0. And minus 0 is just 0, so the pH is 0. So that makes sense in terms of what I see for that original pH. The pH was 0. And then what I did was, again, opened up the stopcock and allowed 1 milliliter to enter, and little by little, as I allow that process to proceed, I saw that the pH was increasing slightly. Now I've graphed pH. This is what on a titration curve, you're going to place pH on the left side, and on the bottom, the volume of the titrant that you add. In this case, it's sodium hydroxide. It doesn't have to be a base, but in this case, it is a base. I'm titrating with a base. Okay? So as I'm adding more and more sodium hydroxide, notice that the pH is rising. Why is that happening? Why is it happening? Well, it's happening because, and by the way, it appears that it's not, that the hydrogen ion concentration is not reducing very fast, but remember, from going from 0 to 1, or from 1 to 2, every time I go 1 increment on the pH scale, the hydrogen ion concentration, or the available hydrogen ion concentration, is decreasing by a factor of 10. So from 0 to 1, and going from 0 to 1, say to this point right here, the hydrogen ion concentration decreased by a factor of 10. And from going from 1 to 2, it decreased a factor of another 10. So from 0 to 2, it decreased by a factor of 100. So what's happening? What's happening is that as I release the sodium hydroxide into the solution, the hydrochloric acid, remember, was dissociated completely, so 100%. So hydrochloric acid in the beaker, or the flask, dissociated completely. As I drop sodium hydroxide, which is also a strong base, it was also dissociating completely to give me hydroxide ions. Remember, hydrochloric acid is an Arrhenius acid because it is producing hydrogen ions. Sodium hydroxide, an Arrhenius base, because it's producing hydroxide ions. But look what's happening. Hydrogen from the hydrochloric acid dissociation and hydroxide from the sodium hydroxide solution are coming together to form water, a neutral solution. So what's happening as I move from left to right, I'm adding more and more sodium hydroxide, and the hydroxide ions are kind of vacuuming up. They're sopping up those hydrogen ions and causing the pH then to rise First a little, but then as we get closer and closer and closer to a certain point, the hydrogen ion concentration begins to decrease dramatically. So I go at this part, in this portion of the graph, the hydrogen ion concentration is decreasing very, very quickly. Now, what's happening as I approach this part of the graph is that the number of hydrogen ions is decreasing because hydroxide is sopping up, is forming water in the reaction that I just showed you. Now, what's happening here? Well, what happens here is that the number of hydrogen ions begins to approximate the number of hydroxide ions. In fact, if I look at this point right there, I can see this point is what we called the equivalence point. Let me write that, the equivalence point. And that means that it is the point at which the hydrogen ion concentration and the hydroxide ion concentration are the same. All right? They are the same at the equivalence point. I want you to notice also that for this strong acid and this strong base, that the equivalence point is happening at a pH, and this just makes sense, at 7. So if I go across, just look across to the pH side of the graph, I can see that at pH 7, that's the equivalence point. And some people ask me, what's the difference between the equivalence point and the end point? The end point is what the experimenter determines is the place to stop the titration. So it might be anywhere from, say, here to here. So remember that the hydrochloric acid had placed into it some type of an indicator, probably phenolphthalein, and when that phenolphthalein turned a color of pink from clear to pink, then the experiment was stopped. So the experimenter then has some leeway, somewhere between here and here, they're going to say, okay, that's enough, stop. We call that the end point, wherever the experimenter stopped. And let me write that down as well. That's the end point. That's where the experimenter stopped. The equivalence point is the exact point at which hydrogen ions and hydroxide ions exactly were equal. Now, also, while I'm talking about the indicator, remember that all through this region, from here to here, the indicator remained clear. So the indicator was clear here, but once I reached that equivalence point, the hydrogen ion concentration began to overwhelm the hydrogen ion concentration. There was more hydroxide than there was hydrogen ion concentration, and this area from here all the way past 1 liter, it was pink. Okay? So in this area here, in this area from here to here, it was clear. From here to here, it became pinker and pinker and pinker until it almost became like a magenta color. Okay? All right. Now, what I also want to talk to you a little bit about is the fact that as I get past this equivalence point, the hydroxide ion concentration, the more sodium hydroxide that I add, I'm just increasing the amount. I'm increasing the amount of hydroxide, and it's overwhelming the hydrogen ion concentration, and the pH cannot do anything but go up and up and up. But of course, there's a limit at 14. I want you to notice also that this point right here, what we call the equivalence point, is 1 liter. So remember, if I started out with 1 liter of a 1 molar hydrochloric acid solution, and I started out with I titrated it with 1 molar sodium hydroxide, well, how much sodium hydroxide will I need in order to completely titrate it? Well, if I started with 1 molar sodium or hydrochloric acid, that's 1 mole per liter, and I have 1 liter, that's 1 mole worth of hydrogen ions. Well, to completely counteract that 1 mole of hydrogen chloride or hydrogen ions, I'm going to need 1 liter of a 1 molar sodium hydroxide solution. So this is the equivalence point here. The 1 liter is the equivalence point. It's the point at which I have exactly counterbalanced the hydrogen ions that were in the flask with the hydroxide ions that came down from the burette. Okay? And that, in a nutshell, is the titration curve for a strong acid titrated with a strong base. So some of the features that we're going to need to know are the fact that pH was equal to 7. pH is equal to 7. So if it's not equal to 7, you know you're not dealing with a strong acid and a strong base. And that the equivalence point came at the point at which hydrogen ions and hydroxide ions exactly equaled. Well, let's talk a little bit about what we might see on test questions. If I were to ask you, is this a titration of a strong acid and a strong base? Well, you see here, first of all, that the initial concentration is not 0, it's 1, but that's okay. It doesn't have to be 0, it's low. We know that because we would have in this container here, in the flask, we would have a low pH because that's an acid, and then slowly the pH would rise, and then at some point, the equivalence point, the pH would be 7, and in fact it is 7. It's right between 6 and 8, so it is 7. So this appears to be a graph then of a strong acid being titrated with a strong base. Okay, these are exactly the features that we would see for a strong acid being titrated with a strong base. But what about something like this? What if I had a diagram that looked like this? In this case, we start at a pH of 14 and go down from there. Now notice that the equivalence point also happens at a pH of 7. The equivalence point is at a pH of 7, but nonetheless, we see the general features of a strong acid, strong base titration, but this time it's a strong base. So I'm adding acid. I'm adding acid. As I am adding acid, the pH is going down. The pH is going down. Okay? So be prepared to answer questions about titration curves involving strong acids being titrated with strong bases, like this situation here, or strong bases being titrated with strong acids, like this situation right here. Remember, the pH in each case has to be 7. If it's not 7, it's not a strong acid, strong base situation. All right, I'll see you on the next video podcast. Thank you.